Here we have a section of double-stranded DNA inside a prokaryote, and we'll observe the process of its replication. Replication all begins at the replication origin, where initiator proteins bind, allowing DNA gyrase to begin unwinding. DNA gyrase is important in that it relieves supercoiling that would otherwise leave DNA too tightly compacted to allow replication. Helicase is an enzyme that breaks the hydrogen bonds in the base pairs between DNA, allowing the strands to separate. Single-stranded binding proteins coat the single-stranded DNA, protecting it and preventing it from annealing. We can see that DNA polymerase III has synthesized a new strand of DNA. Since DNA polymerase III only creates DNA in the 5' to 3' direction, we can go ahead and label all of our 5' and 3' ends based on the principle that DNA is antiparallel. Seeing as DNA polymerase III was able to continuously synthesize DNA, we call this continuous synthesis, which means this must be the leading strand. This new enzyme is RNA primase. It lays down RNA primers on the lagging strand so that DNA polymerase III will have a starting point for synthesis. Here we can see the RNA primers in yellow, distinguishing them from the newly synthesized red DNA and the old blue DNA. They provide the 3' hydroxyl group that DNA polymerase III recognizes and can add nucleotides to. Clearly, this kind of synthesis is discontinuous, since the DNA is synthesized in fragments, making this the lagging strand. These fragments of DNA between RNA primers are called Okazaki fragments. Even though this synthesis is discontinuous, DNA polymerase III still creates DNA in the 5' to 3' direction, so we can still label our ends. DNA polymerase I functions to replace yellow RNA nucleotides with red DNA nucleotides. The RNA primers were helpful to begin synthesis, but true DNA does not contain RNA nucleotides. Now that the primer has been replaced, a phosphodiester bond is missing between the 3' hydroxyl and 5' phosphate of two adjacent Okazaki fragments. This is called a nick. This is where DNA ligase comes in. It browses the DNA for these nicks and seals them. This process will continue until the entire genome is replicated. As DNA gyrase further unwinds DNA, helicase will follow, further separating the strands, while RNA primase periodically deposits RNA primers on the discontinuous strand. DNA polymerase III on the leading strand will follow along with helicase, continuously synthesizing the new strand, while other DNA polymerase III enzymes synthesize the Okazaki fragments on the lagging strand. DNA polymerase I will replace the RNA primers, and DNA ligase will seal the nicks.